Running. And okay, now the recording's running. And we can hear you clearly and see everything clearly, so we're in good shape.
Okay. Thank you very much, John. And welcome. Welcome, everyone, once again, or welcome back for those who have joined us previously. Welcome if you are joining us in our course, One World, One Health, One Medicine, for the very first time. At St. George's University, the year 2017 is a very auspicious one for us, where we are in fact celebrating our 40th anniversary. And as part of the celebration, we would like to engage but also join yourselves who are joining us in terms of sharing our experiences, particularly in the area of One World, One Health, and One Medicine. Today we have our Vice Provost for International Program Development, Dean for Graduate Studies, and Professor of Parasitology, as well as Director for Research here at St. George's University, Dr. Kyle Macpherson. And he himself, as part of his life experiences and the seminar he will be presenting today, exemplifies the One World, One Health, One Medicine concept, where we will be embarking on a journey across the continent of Africa, examining a particular issue that engages both humans, animals, and of course, the environment. So I hope that you guys are ready. I certainly am getting ready as we join in the seminar today from Cairo to Capetown, a reflection of One Health in Africa. Dr. Macpherson, it's over to you, and I'll be back at the end of the seminar.
Thank you, Dr. Bedesi, and I'd like to thank Dr. Bedesi for his vision of putting together this lecture series once a month, which was started with Chancellor Modica in January and Dr. Wendy Crawford last month, and Regan Schwartz, who's a veterinary public health student, will be presenting next month. And it's my great privilege to give the third of the lectures in this lecture series. To celebrate the 40-year anniversary of St. George's University. And I was born and grew up and spent 35 years in Kenya, and I wanted to share with you my scientific journey through the field of One Health. The distance from Capetown to Cairo is 4,478 miles. That the continent is huge, and I just want to put in perspective for some of those who are not so familiar with the continent, just how John, I don't have control over the slides.
Sure, 1 second, let me get in there. So I'm in here now.
Oops.
Okay. Try now.
That's great. Thank you, John.
Okay. Cool.
And so Africa is 1/5 of the world's land surface area. It's currently got 16% of the global population. It has, it's about 3 times the size of the United States. In fact, the US could be fitted into just 5 of the countries in Africa. The largest country is Algeria, and together with Libya, Sudan, South Sudan, the Democratic Republic of Congo, would be the same equivalent size as the United States. So it's a huge continent. If you look at the GDP, though, Africa hardly would register today. This is going to change over the coming decades. It has a wide variety, obviously, of climates, from the Mediterranean climate along the coast of North Africa, the Sahara Desert, which is one of the hottest deserts, through equatorial Africa and the rainforest to the Kalahari Desert in the south. There is snow on the mountains in the Atlas Mountains of Morocco and also in the Drakensberg mountains in South Africa, and on the 2 highest peaks in Africa, Kilimanjaro and Kenya, which are close to the equator. So today there's 1.1 billion people in Africa, and it is one of the youngest continents. 50% of the continent's population is under 25 years of age. In 1950, which doesn't seem that long ago to many of us, there were only just over 200 million people in Africa, now 1.1 billion. And by 2100, in just 83 years' time, there will be over 4 billion people in Africa. And Africa's population, as estimated by the United Nations, is set to change more dramatically than on any of the other continents. This is going to impact, obviously, global health, One Health, particularly Africa. So it's considered the birthplace of mankind. The studies in Ethiopia and Olduvai and Lake Turkana found the earliest hominids. It's had ancient civilizations, which I'll talk about in a minute, and there's over 2,000 unique, many of these languages are unique just to Africa. So the civilizations in West Africa, and obviously in Egypt, where the pyramids are a standing testament to the ancient civilization there. These similar pyramids can be found from a different kingdom in Sudan, through down to the great Zimbabwe ruins in what is today Zimbabwe. At the end of the 19th century, there was a scramble for Africa, recognizing all the natural resources, the potential of Africa. And there were many meetings in Europe, in Berlin in particular, to divide up Africa to the different colonial rulers. France and Britain, Germany, Portugal, Spain took most of the colonies, also Germany. And during the First and Second World Wars, the theater of war took place in some of these countries in Africa. Ethiopia and Liberia were never colonized. Just to show you, the boundaries were drawn up by the colonial rulers and disregarded the existing peoples that lived in various areas. They followed natural courses. And this is the boundary between what was then German Tanganyika and British Kenya. It was a straight line. And the Kaiser complained to his cousin, Queen Victoria, that she had 2 snow-capped mountains in Kenya, and he actually didn't have any. And this is Kilimanjaro and Mount Kenya, the 2 highest peaks in Africa. And so they agreed to redraw the map so that Kaiser could have one of these snow-capped mountains. And today, if you look at the map between Kenya and Tanzania, you will see that the boundary actually is curved around Mount Kilimanjaro. Profound decisions were made on the basis of certain facts. So today's boundaries are jealously guarded by the independent countries that exist today. So I'm going to talk a little bit about Kenya because it reflects a little bit of the history of many of the countries. Railways were seen to be the way to provide transport to go to from the coastline to the interior. And in Kenya, many indentured workers from India were brought in to help with the development of the railways. And the railway was to run from Mombasa through Kenya to Uganda and to the Congo beyond. And the railway was the focus of the development of the interior. Every 90 miles, a railway head was established. So this is the photos from the time, and this is in 1988 is when my grandfather migrated to Kenya. He had actually spent some time in the Caribbean, and he became a sugar farmer on the shores of Lake Victoria. And this is him with a buffalo in the photograph below. So one of the heads of the railway was Fort Smith, and Fort Smith is where the vet school at Capetown is located today, just outside Nairobi. And this is a picture of Fort Smith, Nairobi is a Maasai word. This was Maasai area, meaning cool waters. And this area became very popular and grew very quickly as a center of commerce, mainly for people visiting Kenya. Many people came. This Nairobi is at altitude of nearly 6,000 feet. So it's cool most of the year, and it has a very pleasant climate that is enjoyed by everyone today. So the hunting safaris and the famous people like Theodore Roosevelt based their safaris out of Nairobi. The city grew very quickly and attracted many people, this 1926. And famous people came, and the person in this picture, second from the right, is Ernest Hemingway. He wrote two fantastic books, The Green Hills of Africa and The Snows of Kilimanjaro. The road system came after the railways, and the roads were built, as you can see, using oxen. The roads were very rudimentary. It used to take years. Even as a kid, I remember going from Nairobi to Mombasa on the dirt road. That took many days of journey. So this is my mother. She was born in Kaimosi in Western Kenya on the shores of Lake Victoria. And 10 years after she was born, this young guy's father was born there. And yes, that's Barack Obama. We haven't found a genetic link to the Obamas, but his dad was born in the neighboring village to my mother. This is Treetops, which is where Princess Elizabeth went up in 1952, and her father died that night, and she came down as Queen of England. So we have a lot of links. On December the 12th, 1963, Kenya received its independence, and Mzee Jomo Kenyatta became the first president of an independent Kenya. Independence came quickly to most of the colonies. Egypt and the Union of South Africa, in fact, were independent since 2010. And the latest country to become independent is South Sudan, which received its independence in 2011 and became separate from the Republic of Sudan. So Africa contributes very little of the greenhouse gases today. This will obviously change in time. It's got the longest rivers. It's got the largest span of waterfall in Victoria Falls, second largest lake in the freshwater lake in the world. And it has much of the world's raw earth minerals that are left. And so there are still huge amounts of natural resources in the country. Moving on to One Health. One Health is a continuum of veterinary medicine and medicine, and there is no real dividing line between the two. And if you take the basic sciences, the delivery of both can comprise One Health. And when the vet school was started here, we actually taught the vet students and the medical students in one class. And looking at the use of animals, they have taught us a lot about cancers. They share same problems with narcolepsy and many other disorders and infectious diseases. If we combine one medicine with one health, the broader connotation, looking at how healthy people, healthy environments, and healthy animals are all intertwined, and the professionals that work in these areas need to work very closely together with climatologists, environmentalists. And this concept was put forward by Virchow, by Sir William Osler, and by many others, Jenner and by Calvin Schwabe, who coined the term one medicine. It's actually a concept that is over 2,500 years old. Hippocrates wrote about the health in areas with different waters and airs. and seasons. And so the whole link between the environment and our health has been known since the first writings of scientific scholars. The World Health Organization records a number of neglected tropical diseases, from viruses through bacteria, nematodes and protozoans, and there's a number of zoonotic infections, and the one I will be focusing on is Echinococcus. So if you look at zoonoses, a zoonosis is defined as those infections and diseases naturally transmitted between vertebrate animals and humans, and they share the same environment. And how is these diseases shared between these groups, and in what environmental situations? One of the great parasites that do link up all these three areas is Echinococcus. And I shall be confining the rest of my presentation to one species, Echinococcus granulosus. It's found on all the inhabited continents. It's a small tapeworm, only half a centimeter long. It contributes many millions of dailies, disability-adjusted life years, and to that has to be added the loss of production from animal production. It's a parasite of canids. Dogs are the main definitive hosts, and they produce eggs which infect sheep and other livestock. This is where I have had, in yellow, the privilege of working in Africa, all these countries, and this is where Echinococcus exists. I'm not sure who is following who around, but I know that I chased the Echinococcus all over Africa, and I'd like to share this with you. So it's a cyclozoonosis. That means it cycles between herbivores, which develop the larval stage, and dogs, which are the definitive hosts. So dogs scavenge livers and lungs, which contain the cysts and protoscolices, and they then develop the tapeworms, and the tapeworms then produce eggs, which are infected with the herbivores. Humans get accidentally infected when they ingest the eggs from dogs. So let's have a closer look at this. Dogs ingest the protoscolices, the larval stage. It goes to the small intestine. Here it grows into an adult worm. This is the intestine of a dog with thousands of little tapeworms. And these little tapeworms then produce segments which are full of eggs, and these are shed in the environment. And in this dissecting dish, you can see the size from the best beer that you can buy in Kenya, Tusker, much smaller than an elephant's head. These little tapeworm segments that are Echinococcus worms that are passed every 2 weeks in dog feces containing lots of eggs. Sorry, Jonathan, I've lost the ability to move.
Okay, let me get.
Okay, thank you. And these proglottids pass out in the, this is the anus of a dog, and the dog licks this area, spreads these eggs all over its coat, and this is a fecal mass from a dog. The proglottids move away by peristalsis, away from this zone of repugnancy, and they get onto the grass, and they sit there waiting to be eaten by a herbivore. Still can't change the slides. John? Hi, John?
Yeah, you're not able to click? I'm here.
Yeah,
you can't click in advance?
No,
okay, sure thing. Yeah, we just got to make sure that it's on the PowerPoint.
Okay.
Make sure the mouse is on the PowerPoint and not on go to training or anything else. And I'll keep an eye on it. But go ahead.
Okay. So humans get infected accidentally by ingesting these eggs, and that's by human behavior. And also, so if you have close contact with dogs, this is a Turkana people, you can get these eggs which are passed out continuously, and many millions of them all the time. They're immediately infective in the environment. Pastoralists that herd their sheep and goats in contaminated areas, the livestock will also get infected. So let's take a closer look at how these eggs, so this is, on the outside is an embryophore which protects the eggs from the environment, not very well. So if transmission is not quick, then the eggs will perish. And the little larva, the oncosphere, is inside the egg. And the larva then, once it reaches the small intestine, pops out, is called an oncosphere, invades the small intestine, and then is passively transported by the mesenteric veins to the first filter, which is the liver. Here they become trapped, and over a period of months to years, they develop into these fluid-filled cysts. And this is the liver and lungs of an animal which has got many such cysts. And this is me approximately 40 years ago with a goat's kidney, liver and lungs showing these hydatid cysts. So if you take a cross-section of those cysts, they comprise of a layer against the host and hundreds of thousands of protoscolices which are contained within these brood capsules. In humans, because the liver filters out most of these oncospheres, the cysts grow mostly in the liver, and they grow very quickly, the Turkana kids. If it bypasses the liver, goes to the lungs, gets trapped in the capillaries in the lungs, this is an example of a lung cyst, but it can be spread to any organ of the body, causing unilocular blindness, and this is a cyst in the brain. So it really has a significant public health impact. Remarkably, this little protozoan parasite that is in the cyst, the protoscolex, if you inoculate it into the peritoneum of mice, it grows into hydatid cysts because of its location. Yet if you feed these protoscolices to dogs, it grows into a sexually mature adult tapeworm, which then goes on to produce eggs. So this parasite, this little protoscolex, has the ability to differentiate into an asexually producing hydatid cyst or into a sexually reproducing adult tapeworm. This has profound implications for surgery, because this is a patient on the table called Larotte. She came in, she was 18 years old, she was an outcast from the tribe because they thought she was pregnant, she wasn't married, and she had a unilocular cyst. The surgeon removed it, and on the right is her 4 years later with multiple inoperable disease. So her abdomen would have looked like those mice, full of secondary hydatid cysts. So my journey with this parasite started almost exactly 40 years ago, just like St. George's University, so both kind of celebrating anniversaries. And I was at Sheffield University. We didn't take parasitology in the zoonosis, the zoology department. And my father visited and said, "What are you going to do when you finished?" I said, "I thought of going to medical school." And he told me about a Rotary scholarship, and it sounded kind of interesting. And we were walking along the shops, and there was a bookshop, and I said, "You know, I'm really interested in parasites." And he bought me this book, Introduction to Animal Parasitology by J.D. Smith. So I wrote to J.D. Smith, who was at Imperial College, and asked him whether I could come and take a PhD studying cysticercosis in the cattle in Botswana. And he said, on two conditions: one, you have to get a first-class honours, and two, you need to get funding. Both of which I thought were pretty challenging, because Sheffield had not awarded a first-class honours degree for 7 years, and where do you get funding? So I did apply for a Rotary International Scholarship, which I was fortunate in obtaining. And about a week after joining Desmond Smith, I appreciated that he was one of the world's leading parasitologists, and he had been a mentor to many individuals like William C. Campbell, who 2 years ago won the Nobel Prize for Medicine for his discovery of ivermectin, amongst many others. And he had a great sense of humor, and he was chuckling at in his laminar flow hood. He was always happiest when he was working in the lab. And he said, you know, I've been working on this thing for nearly 40 years, and the more I understand it, the more I don't understand it. And the same is true for me. So 40 years later, it's still quite an enigma of how we control and conquer this parasitic disease. But a week after I joined, Sir Michael Wood, who founded the Flying Doctors in Kenya, the African Medical and Research and Education Foundation, came to see Desmond Smith, who was the world's leading authority on Echinococcus. And he said, I need somebody to come and help me because 35% of the patients we operate on come back with multiple inoperable disease. And Desmond Smith says, well, I've got a young Kenyan here who might be somebody you could work with. And I met Sir Michael, who was my absolute hero growing up in Kenya. The Flying Doctors was an institution that everybody looked up to. And so I changed from cysticercosis to echinococcus in about 30 seconds. And this is Sir Michael on the left-hand side operating on a patient with Hydatid disease. This is a cyst that's infected. On the right is a cyst that's been removed by Dr. Gilchrist, and the prognosis for the patient is pretty good. But this is a very complex parasitic infection. This is a cyst with many different daughter cysts, and Sir Michael is removing these daughter cysts, ladling them out. And you can see how leaving any of these daughter cysts or the protoscolices inside these daughter cysts in the tens of thousands would result in secondary multiple inoperable disease. So one of the visitors, and we had a number of visitors, that is another mentor, is Sir Martin Wood. Sir Martin was the head of Oxford Instruments. He was the founder of Oxford Instruments. And one of the problems was the surgeons could never tell what they were going to go into and find when they did a laparotomy. It was always a surprise because there were no imaging techniques at that time. And I remember Sir Martin said, don't worry, Cal, we've worked things out. All the tissues in the body have different magnetic resolutions, and we could easily develop a new device imaging the body using magnets. And I said, yes, you must be crazy. Anyway, the person who thought about, conceived the idea of MRI. He didn't win the Nobel Prize for developing it because that was developed by 2 other people who did win the Nobel Prize. But he had the idea, and he was knighted for that. So I joined the Flying Doctors in 1978, this is Michael, to work on cystic echinococcus, and received a Wellcome Trust Scholarship as well as the Rotary Scholarship to conduct the work, and flew with Sir Michael Wood, a nurse and anaesthetist, to many remote rural hospitals around Eastern Africa, Somalia, Ethiopia, Sudan, South Sudan today, Uganda, Tanzania, and in Kenya, in these little 4-seater planes, serving these hospitals that didn't have physicians working at them. My main role was to work out why echinococcus was so common amongst the Turkana people in Kenya. And these are the Turkanas. They live in these acais, and they move as the need for grazing takes place. They're wonderful, independent-minded people. who are very strong-willed, and they depend on their animals because they only drink milk and blood. Animals are used for much more erudite reasons, for cementing friendships, for bride price, than for eating meat. So I received all my permits. I was registered at Imperial. I was also registered at the University of Nairobi with Professor George Kanoti, and I worked with the AMREF to work on this disease. We became very nomadic. When you're following nomads, if you want to understand the epidemiology, they have their movements and their seasonal migrations, and we had to move with them to understand how transmission was taking place. You can imagine, we have a few stories of crossing rivers and waiting for flash floods to go. And if you didn't take the fan belt off your Land Rover before you crossed a river, you would end up like this and waiting for the river to go down. Similarly with airplanes, and you can see Michael Wood is also pushing the plane out of the mud. It was a harsh environment in which to work. But the epidemiology was studied with many students coming to help during the holidays to look at how women had 3 times the disease prevalence. So women stayed at home and were concentrated in these acais with their dogs. They also cleaned the babies, and so there was a very close association between women, infants, and dog. Whilst men went off herding their sheep and goats and weren't as closely linked to the dogs, and they didn't use dogs for herding. So these are also food preparation, lots of dogs you can see, and dogs accompanied women most of the time. We also showed that very few animals were slaughtered, but when they were, they didn't inspect the meat, and if cysts were found, they were fed to dogs. The other great problem was the droughts, which occurred every decade and now are occurring ever more frequently. And the current drought in Somalia and South Sudan, northern Kenya, and across to Uganda is just a number of droughts that have taken place in this area of Africa. And it really does help the propagation of the life cycle of Echinococcus, because the animals will die, the dogs will feed on the animals, get heavily infected, the dog population rapidly increases, the drought subsides, new younger animals are brought in to replace the livestock that were there. They're very susceptible, the dogs are heavily infected, the dogs then slowly die off. It's a harsh environment, they live for about 1.5 years. And the animals are infected, and the cycle has repeated over these decades. Another wonderful mentor, very inspirational individual was Calvin Schwabe, who coined the term One Medicine. And Cal was the first head of the newly established veterinary public health department in the World Health Organization in 1974, and here he is. And he would visit us in Turkana. He had a passion for Echinococcus, and he had a passion for the nomadic peoples. He wrote a number of books in addition to veterinary medicine and human health. He wrote one on cattle, priests, and progress in medicine. And he would revel in cooking. We had a lot of spitting cobras around the camp, and he would fry these up. And we had an animal house with rodents, and he would make rat soup and spitting cobra steaks for dinner. And these you can find in other great recipes in his book, Unmentionable Cuisine, available on Amazon. But he was an inspirational visitor and another mentor. And others were 3 veterinarians, one from Australia, Jack Arundel, from the UK, Peter Stevenson, and Lars Karstedt, who was from Canada. And excuse me, my supervisor, Desmond Smith, did tell me when I presented my proposal to him, if you do 2% of this work you propose to do, you'll have done a fantastic PhD. How are you going to examine wildlife? How are you going to capture hyenas and jackals? I said I didn't know. And these are the people, the veterinarians, that made it happen. I was very fortunate. We examined wildlife of many species. Last casted, and I caught hyenas and jackals. We experimentally infected them with the parasite, and almost everything that we set out to do was accomplished through their collaboration. And the lab studies were really to look at the strains, and we now understand that Echinococcus granulosus has many different genotypes and host assemblages, which is much too laborious to go through here today. But much of my PhD was focused on lab studies on the biology of this parasite. But we also, the Turkana are not buried. They leave their dying out so that they can be eaten. How do you get to heaven if you're put under the soil, is their philosophy. And so we took human material from operations and fed them to jackals and hyenas to then harvest the eggs and then infect camels, cattle, sheep, goats, and also baboons and rodents and vervet monkeys. So these studies, looking at host assemblages of this parasite, formed a lot of the work and were sponsored by grants from the Wellcome Trust and by ODA. So then came the evolution of ultrasound. And ultrasound was a hospital-based imaging technique until the development for the veterinary world for diagnosing pregnancy in sheep and pigs. And in 1980s, ultrasound was further developed. And this is a study I did with Ellen Ratner in South Sudan, and you can see the techniques today use very small transducers, very small portable equipment that you can take anywhere in the field, and the knowledge of physics and the physics of sound coupled with the advances in computers have revolutionized the role of ultrasound. So my own experience with ultrasound started with my baboons and vervet monkeys. They were eating me out of my Wellcome Trust grant. I didn't know they were infected. There was no way of telling. We took monthly blood samples. The results were a little confusing. And then a lady I knew was diagnosed as being pregnant with this 1 centimeter fetus, and I went to see the OBGYN lady, and I said, would you mind screening my baboons? And she found this an unusual request, but she granted it. So we anesthetized the baboons, wheeled them through on a wheelbarrow, and she found the cysts. And this is one of the cysts from one of the baboons, and thereby started another chapter. So I was sent. Sir Michael Wood said he did all the operations. The anaesthetist said I don't do diagnosis. The nurse said I'm too busy. So I was sent to learn how to use ultrasound, and these are early pictures. And Sir Michael Wood was fantastic with his support for this. And we did over 600 operations where he would see the image, we would discuss what the operation was going to be about, and then collected the material from the operation. And along with Anne Story, who was a French Jew who had been at Auschwitz, had survived, she learned to fly at the age of 65. And this is her plane, Alpha Zulu Tango, which is many decades before AZT for HIV. And she got very enthusiastic and helped me by flying me to huge areas in East Africa to do ultrasound surveys amongst very remote rural populations to find out how prevalent this disease was in this whole area, which had previously been blank on the map of Echinococcus. Our surveys in Turkana also turned up a number of patients, and so these patients went to Liberia for treatment with a new drug called albendazole. And then the Beecham gave us a large grant to start our own albendazole village in Turkana.
Dr. Macpherson, I'm gonna pause you for a second. We've got someone who's on a phone right now that their audio is coming through.
Okay.
I can't mute them from this side. I dismissed someone on a phone, but I am having trouble muting on this side. So everyone, especially if you're on a phone, just make sure that your phone is muted or your microphone is muted. It sounds like we've still got it. Is someone on the phone there? Can you mute that, please?
Alrighty.
Okay, Dr. Macpherson, keep going. I'm gonna work on dismissing this finding and then dismissing this attendant. But go ahead. Thanks.
Well, here's that patient.
Hello. Yeah.
Yes, I can hear you. All right, I will continue. So here's that patient that I showed you, Larotte, and she was one of the first patients to receive albendazole. She was treated and cured, and she still works at the Hydatid Center in northern Kenya. Serology used to be the gold standard for diagnosis, but it is unpopular. As you can see, the mother covering the face of the infant, very low predictive values, and it is used as a confirmatory test. So 1982 was a watershed year. It's a year that WHO called for a meeting on the immunodiagnosis of cystic hydatid disease in Geneva, and they invited me to give a paper at the very end of the meeting on the role of this technique, ultrasound. Albendazole trials had started, and with ultrasound we were able to look in vivo at the effects of this drug. And the government of Kenya said enough research had been done, and so they wanted to have a cystic echinococcus control program implemented so that the highest rate of echinococcus in the world could be controlled. So to really illustrate the problems with serology, besides its unpopularity, the sensitivity is a property of the test, as is the specificity, but the predictive value of a test depends upon the ability of all the positives that come back from a test as a proportion of all the positives gives you the positive predictive value. And when you get low prevalences of a disease, you then have a very low positive predictive value. And you can see here, no test in those days was 100% specific or 99% sensitive, but at 1% prevalence, the positive predictive value is still only 50%. So just a little bit better than flipping a coin. So these were the headlines at the time. Hydatid control program was to be started. Historical facts. And I'll just quickly go through the Turkana scenario. So in the 50s, a few patients walked to Kitale Hospital, the nearest hospital. There were none in the district, and they were very large patients with very large cysts. They'd walked 200 miles to seek treatment. Calvin Schwabe then estimated the incidence of the disease in the mid-60s at 40 per 100,000. Our first serological tests showed about 220 per 100,000. And then when ultrasound came in, we showed the true nature of the disease. And so the control program was started in the most endemic part of the district which was in the northeast corner of Kenya. So another mentor was George Nelson, who was the director of the Liverpool School of Tropical Medicine. George had gone out to East Africa after he had finished his Doctor of Tropical Medicine and Hygiene degree and described the first cases of Trichinella south of the Sahara, and it's now called Trichinella nelsoni. And George was the physician consultant who oversaw the whole of the control program from the Liverpool School. Another person was Lord Lawson Salisbury, who was very instrumental and very influential at helping us with our control program. And this is a picture of Lord Salisbury with the charter and the second class of the School of Veterinary Medicine in Trinidad, and Lawson was my external examiner, and he more recently became the second president of WINDREF. Lawson Salisbury's been an inspiration to many in the field of zoonoses. So the control program began. The Kenyan government appointed me as the director of the National Pilot Control Program, and with AMREF as a partner, the program began. We started by building our very impressive research institute. There were 2 PhD students from Germany, because GTZ funded a lot of the initial part of the project. Eberhard Zeli, who until recently was with the project, he's now retired, and Thomas Robig, who works in Honnenheim in Germany as a professor there, and myself. So we had a 3-bedroomed research institute full of scorpions and spitting cobras. So this was the institute, and this is the village. You can see the circles down here of Lopeding. It's 5 kilometers from Lokichogio, which is on the border between South Sudan and Kenya. And you can see the research institute buildings there, and halfway between was the water hole. And it's a semi-desert area where water is very valuable. So I was on my way up to Turkana, and I stopped at a mission station to spend the night, and the missionaries were leaving and said, "Do you want to buy a waterbed?" And I said, "Well, are waterbeds cool in the desert?" And they said, "Oh, they're very cool at night." So I said, "Okay, I'll buy this waterbed from you," and it's on the roof rack of the Land Rover there, and I transported it up, and then I used 200 of the Turkana, the whole village from Lopeding, to pass jerry cans from the borehole, which you can see here, which is how we accessed our water, to the waterbed, and the empty ones going back. And it was profound, the changes that took place over the next weeks. All of a sudden, the elders, like this is the witch doctor Latom and Loily, each of those scarifications tells you that he had killed somebody, and it's a mark of respect, said that I was a very wise man. And I said, "So why is this that I'm now a wise man?" He said, "Well, in our part of the world, water is the most scarce resource, and you actually sleep on it. So when you get thirsty at night, you just take a straw and take a sip of water from your bed. We think this is incredible." And so just by accident, this helped with the perception of reality with the people. And the control center was built thanks to large grants from the Australian government, amongst others. This is myself with the chief and all the Turkana who protected us. This is my lab staff. We had a team of about 30 people. And this is the main team, was my educators, Henry, Edward, Lorraine, and the chief man was Carling. And all of these people had many scars indicating they were fierce warriors in the area. I needed to have the baddest of the bad working with me, because this was an area with a lot of cattle rustling, And just like in the US, you have football where you have people coming together and you look at these people going attacking each other on the sports field. In Britain, we have the soccer. And in this part of the world, it's these cattle raids, and they steal each other's cows. And this is Lorraine, who was shot by a high-powered bullet, survived because one of the Flying Doctor's team arrived the next day and was there to help him. So predicting whether to undertake a cattle raid to South Sudan or to Ethiopia or Uganda, to the Karamojong, was based on the witch doctor, Latom, predicting whether it's a good time or not. And interestingly, he would look in the mesenteries of goats and sheep, and if there were a lot of blood spots, this meant it was a good time. And these blood spots, as we now know, are caused by Haemonchus contortus infections, which is one of Lord Salisbury's main parasites that he worked on. It's a great place to bring up my two sons, who were born in Kenya and grew up with the tribe. This is Murray, who's now a graduate of St. George's with his MD. I was changing the oil on the Land Rover, and I think he was trying to turn himself into a Turkana. So we started with education. You can see most of the population were women. We had one message: don't feed cysts to dogs. And a few years after starting the program, Red Bana, they Save the Children from Norway and Ethiopia, invited me to look at the Nangatom and the Dasanech people, and they started singing a song, and I said, so what are they singing? And they said, well, it's a little silly. And I said, no, what are they singing? And they said, well, they're saying don't feed cysts to dogs. And this had been translated into 7 different languages and quite accurately across international boundaries. So we spayed all the females, and this is Timothy Washira, my first PhD student. He's now the Vice Chancellor of a theological college in Nairobi, but he is a veterinarian, and he treated dogs every 6 weeks with praziquantel. So we came up with this great idea of collaring the dogs once they were treated. And I used to take lots of students from the UK mostly, and they would look through the binoculars. If the dog collar needed changing, they would run after the dog, because the dogs recognized the treatment Land Rover very quickly, and they would race off and the students would catch them and treat them. But after a few dosing rounds, we found that the people actually liked these dog collar discs, and they wore them, and so we gave all 10,000 away and had to abandon that project. So by the end of 1987, we had a control program that was running very well, and I moved on to the Swiss Tropical Institute in Tanzania, first as deputy director and then as director, to look after malaria and lymphatic filariasis programs. Just want to mention a few people we met along the way whilst I worked with Sir Michael. One was Mary Leakey at Olduvai, and we stopped and spent some times with her. The other one was George and Joy Adamson. And down below in his bath is Tony Fitzjohn. There's a wonderful documentary on him. He was a lorry driver, took people transcontinentally, and he broke down and he stayed with George Adamson to look after his lions, or Born Free fame, and he stayed for 18 years. And Tony then was invited by the government of Tanzania just before George was killed, and he set up Mkomazi. And I mention this because Mkomazi is a fantastic 3,500 kilometer national park. It's the latest national park, established in 2006, and is a great extension of the Tsavo Mkomazi Game Reserve, which is facilitating the growth of the populations of rhinos and also of Cape hunting dogs. So interesting people. So over the last decades, I have been interested to find out why in hot, dry environments that are mostly inhabited by Muslims, that this parasite occurs in the high prevalence that it does. In the Hadith and in the Quran, there are sayings that angels will not enter a house where there is a dog, and when a dog licks from your vessel, you must wash it 7 times. So I was invited to Libya and to Morocco and other places to do ultrasound surveys. And I just put some of these up. This is Mohammed Shambesh screening a baby. And just to show how kind ultrasound is, you can use it on sleeping babies. And you can screen pregnant mothers, which attracts the whole population to the surveys. Libya, by the way, has some of the best Roman ruins in the world and some of the greatest examples of Roman sanitation, which must have been pretty spectacular during the Roman era. In Morocco, this really demonstrates the One Health nature of echinococcus. We had veterinary students, medical students, radiologists, politicians, and environmentalists who would work with us on these surveys. So a lot of surveys have been done. This is not a complete list, but a show that tens of thousands of people were screened, and a number of things were found. One is you can calculate the public health importance of the disease in an area from the case mix. You can also use questionnaires and also use the local physicians who are going to treat all the patients that you find to show them how asymptomatic a disease is. And this is in Morocco. We would go to the slaughterhouse, we'd get the hydatid cysts from the sheep and goats, and we'd ask, have you seen these types of parasitic lesions before? And if you have, what did you do with them? But just by asking, before they were screened, have you ever had an operation and what was it for? And they would bring their operating records. We discovered that lung cysts were very common throughout all ages, and liver cysts were more diagnosed in older ages, so showing the less quick growth of these cysts in lung cysts. Dogs, you would ask what is the role of dogs. In some communities, like this one in Uganda, dogs were used for herding amongst the people in Queen Elizabeth National Park, where I had the pleasure of taking a number of vet students over the years to look at hyenas and lions and leopards and their role in echinococcus in that part of the world. I took my daughter Maggie with me, who's now in pre-vet. This is Dr. Butler, who came on one of the trips. He then went on to do his PhD at UC Davis and his residencies in Columbia, and is back on our faculty, and we're very pleased to have him. And he had many interesting visitors during his camping time in Queen Elizabeth National Park. If you ask the question, what do you do with these cysts? And most of the people would say, feed them to dogs. You could also find out that this disease was an accidental infection, and the longer you lived in an endemic area, the more chance you had of getting infected. We worked on the natural history of the disease, and this is the WHO classification, and I was asked in '94 to chair a WHO committee because there were many classifications, and the first one was started by the gentleman on the right, Hassan Gabi from Tunisia. A clinical one was proposed by Karamani, who's in the middle there, and the three of us worked with about 60 other radiologists from 16 countries speaking about 8 languages on this classification. And we changed Gabi's classification by just moving CE2 and CE3. This was published in the WHO and Acta Tropica. We published a lot of papers along the way, many in the Lancet and in other journals. it's quite interesting to reflect how the mentors, Calvin Schwabe, George Nelson, Lord Salisbury, influenced the journals that we published in at that time. Also edited a book with Francois Meslin, who was the chair of the WHO's Veterinary Public Health Department, and Alex Wendler, who was the head of rabies in Canada. And just to remind you, there's not too many shopping days to Christmas, so you can buy this wonderful book. It has some great properties, and I just want to illustrate one. This is Trevor. I gave him a copy of the book, and I asked him if he would have a quick review. I went back into the office to see what he thought of it, and here he was. So if you suffer from insomnia, this book may be a cure for you. So just to come full circle, back in 2007, St. George's University awarded the AMREF 2 free scholarships for 2 students to come to study the MPH degree here at St. George's University. And this is myself with Michael Smalley, the Director General of AMREF. And he was very kind. And the person who facilitated this in the red cardigan there is Victoria Kamotho, who was one of the students. And I asked her if she would help set up a selective in Kenya so that more students could go out and visit Kenya. And since that time, we've run a selective. This summer, we'll be taking 2 groups of students, that's 40 medical students, to Kenya to work with the Maasai, to visit the Maasai Mara, and to see how healthcare is delivered in Kenya. One of the students who came a couple of years ago, who went to the Keith B. Taylor Global Scholars Program, was Nadia Solomon. And Nadia asked to do an MD MSc degree. And I said, well, AMREF had just asked me to evaluate 30 years after the control program, or 35 years, what the program had achieved. And I said, well, you could review the data and use this for your thesis. And the first thing that had changed since my time was you had to go through very stringent ethical review boards. This was to review the records that had been collected many decades before. And she did a fantastic job, and the publications from that have already started to appear. What she found was that the control program has had a profound effect, reducing the number of younger people who are infected, and also the sizes of the cysts have got smaller and smaller. So they're younger and younger. There is still some transmission, but the control program has been a great success. So I just want to end with a quote, since this is Cairo to Cape Town, with the person who was in this island for 27 years, who said, education is the most powerful weapon with which we can change the world. And that, of course, was Nelson Mandela. I'd like to thank all the mentors. It's been a great 40 years experience. And Michael Wood wrote up the Turkana chapter in his book, Different Drums. And so I'd like to thank all the people who I've worked with over the years, and thank you for listening. Thank you.
Alrighty then, that was an exhausting trip as we engaged.
4,000.
Yeah, exactly, a couple of thousand, 4,000 plus miles. But thank you very much for sharing your experience with us and ensuring that the conversation on One World, One Health, and One Medicine very much continues for the benefit of our learning community that we have here now. And having received your very much relevant and interesting seminar presentation, we'd like to open the audio, we'd like to open and invite anyone with questions, with comments, and I'm certain that many of you have some burning questions or even some comments that you'd like to share with us. And John, if you can help facilitate that, we appreciate that, and let's continue the conversation.
Thank you.
For sure, yeah. This is John talking. So we have a couple notes of appreciation in the chat. What I'll do is I'll have anyone that wants to ask a question raise their hand using the little hand icon, and I'll unmute you and we'll ask questions that way.
Thank you. If I may ask you a question, while we wait on other questions, if you were to identify, besides hydatidosis as a disease, what other public health burden also exemplifies the one health, one medicine type of theme in the continent of Africa?
Well, I think that all the neglected tropical zoonoses, perhaps the most underestimated one is Toxocara. Toxocara is a nematode parasite which causes 4 syndromes in people: covert toxocarasis, ocular toxocarasis, visceral larva migrans, and it is very underdiagnosed, causes neurotoxocarasis as well, is very common. Another one is probably toxoplasmosis. Perhaps a third of the world's population are infected with toxoplasma, which we get from eating undercooked meat, and secondarily from cats which produce oocysts. And both of these, because of their ubiquitous nature and the high prevalences that you find, the effects of the environment on their distribution, the human behavior, the fact that Africa's population will quadruple over the next 80, 90 years. If we don't take care of the environment in which the populations are going to grow and evolve, the more that we live in close proximity with our animals, the greater these issues are going to become. And so we really have to work on putting in the public health infrastructure to try and mitigate the effects of these problems.
Okay, thank you.
Thank you. Yeah, I'll unmute for the next one, but we have a question coming in from the chat from Brian Edwards. And he's asking, why was hydatid disease so prevalent in Muslim-predominant areas?
That is a tremendous question, because it is something that we could not understand. There wasn't this close association with the dogs. The areas that we found it most commonly was in the pastoral people in Morocco, and we feel that the transmission is completely different. It's influenced by contamination of water sources or food sources, and so you get indirect rather than direct contamination. And because the environment was cooler, the eggs of Echinococcus in Turkana would be dead within about 30 minutes when they're passed, if they're not ingested by an intermediate host. But if you go into the Atlas Mountains and in other places, the environment was a lot cooler, and so the eggs survived a lot longer. And it was similar in parts of Libya and other areas where you've got most likely indirect transmission and contamination of fruits, vegetables. It's probably going to be important areas. Quite interestingly, the dog population was not controlled as well as we thought it would be. Dogs were allowed to exist, and you would get a lot of semi-owned dogs, stray dogs. So the dog populations per se were not very different. And so you had quite an environmental contamination that would have existed. And some of the festivities, Eid, where a lot of animals are slaughtered on one day. Yeah. A lot of dogs would get thrown cysts. So a lot of home slaughter, which resulted in dogs getting infected. And so inadvertently the propagation of the disease. But it is quite a different epidemiological setting.
I have a question. I was curious to know, as you were talking, what the rules are around infecting animals with parasites, and do you have to get permission for that kind of thing, or not?
Well, in those days, not. What I had to get permission from were the vet school in Kibeti in Kenya looked after my hyenas and jackals and also the sheep, goats, and cows. I kept my camels on a farm in northern Kenya, which didn't have camels, and the farmer had to be convinced that if I introduced this parasite into the camel, that it wasn't going to get into his cattle, sheep, and goats and into the wildlife. And so that was a lot of negotiation with the owners, and they were always present when we post-mortem the camels subsequently, about 2 years after the infection. And some of these camels were incredibly heavily infected. And so we would take all the material from the farm, and we were very seriously quarantined when these results were recorded. So the process today would be obviously through very strict institutional animal care and use committees, but it was a way to investigate whether non-human primates, and I should really thank people like Richard Leakey, who was the director of the Institute for Primate Research in Cairo in Nairobi, who allowed me to infect, and with some of his workers, and I'm still in contact through the wonderful world of ResearchGate with people like Suleiman, who was a veterinarian who looked after the baboons and the vervet monkeys there, and we published the papers together. So this was, in my opinion, the best way to demonstrate whether a strain was infected to another host, or whether there were physiological barriers to infection. Today, much more use obviously is made of molecular markers and using advanced biochemical techniques and biological techniques. But sometimes we're also losing, when we identify molecular differences, what that actually translates to in reality. So these are studies that I think over time the results will marry quite closely.
Interesting. Okay. We have a question from Motunde, and it's a long question, so I'm going to try to just unmute you, Motunde, and have you ask it. So, Motunde, you're unmuted. If you want to ask your question, you can go ahead now.
Hello, Mutunde. Can you hear us?
I'm not seeing his audio trying to come through, so I'll read the question. I'll give my best shot here. So Matunde was asking, with the world's focus on climate change, what do you think may be the impact on hydatid infections and other neglected tropical diseases in Africa, and what are the steps, if any, being done in that regard?
That's a great question. Obviously, predicting the future is very complicated. However, there are climate models showing the changes that are going to take place in Africa over the coming decades. And just like there have been profound changes, the Sahara Desert used to be an incredibly fertile forested area of the planet. And that's why under the Sahara Desert there are enormous aquifers which Libya exploited to provide water for the coastal towns along the Mediterranean. And places like Libya has enormous, and Algeria, oil reserves, because these were areas that had incredible big rainforests. And So moving forward, the predictions of how the environment will change in Africa is very interesting to look at. And I didn't include some of the data here. We don't know how accurate it will be, but certainly these droughts that used to occur with regularity are going to increase. I think climate change will have to be taken into account. If you look at Lake Chad is now a tiny fraction of what it used to be. And many other areas have already changed dramatically, just over the last 20 to 30 years. With Africa's population set to grow so considerably, it is likely that the whole rural-urban migration that has been taking place all over the planet, and now with more than half the world's population living in urban areas, that the climate change might not affect us as much, because if most people are living in urban areas, the transmission of this disease will continue. And if there are these extreme droughts where most of the livestock perish, then I think these cycles will continue. But that's a great question, and it's a very difficult one to predict with certainty.
And even in terms of vectors of disease as well, as vector populations expand both longitudinally, latitudinally, and altitudinally as well, where we're seeing the emergence and re-emergence of many vector-borne transmitted infections as well.
If you look at vector-borne diseases, that's a good. There's a place called Amani in Tanzania, which was a malaria research center set up, I think, in the 60s or late 50s, above the malaria transmission zone. And because it was at altitude on Kilimanjaro, and today malaria is transmitted at Amani. And so the vectors, because of the climate warming up, are able to migrate vertically. The seasonal changes, the vector-borne disease burden is going to increase. And now, with the very worrying development of Artemether resistance by the malaria parasites, and us not really having new drugs to combat malaria, and the new potential zoonotic viruses that are still waiting for us. It will be very interesting to see the changes over the next few decades.
Certainly requires for us to place significant emphasis on the One Health, One Medicine concept as well, because that's understanding the problem as well as advancing solutions also.
And taking into account the cultural differences that we have. I mean, with Ebola, there were 22 outbreaks of Ebola. And they were all in little villages and contained very quickly. And, between 100 or 200 people died. And then there was this outbreak in West Africa where cultural differences played an important part in the spread of the outbreak.
Yeah. Thank you for the question, Matunde. So the next one that we have on the list comes from Brian Edwards, and we'll try again. Brian, if you're on, I'm gonna give it a shot to unmute you and read that question if you like. I've unmuted from Brian from my side. You've got to hit the little microphone until it turns green, and then you'll be on with us. Well, if you get it, Brian, we'll get you on here. But it's not a good day for a microphone, so I'll read it again and bear with me because it's another long one. But so the question that comes from Brian is the map of prevalence of CHD, which I assume was one of the slides that you showed. It seemed to show that the USA did not have any cases because of the temperature and climate places such as Miami or southern Texas seems that it would be susceptible to that disease. So is there a particular reason why this disease is not spread to these areas?
The history of Echinococcus in the U.S. is very interesting, and it was very prevalent in California amongst the Basque people who migrated there, and they used dogs in a transhuman style of herding their sheep. And it was very common around the Four Corners, and it was eliminated through primarily education. And so there was a big move to educate the farmers to treat. And so in the US, he's nicely observant, and if I put Alaska in there, Alaska does have cases of a related disease, Echinococcus multilocularis. But granulosis is mostly seen in immigrants who come from endemic CE areas, and they might be driving and they might have an accident and they suffer an anaphylactic shock from their cyst being burst or something. So it's found quite incidentally. And at the moment, as far as we know, there is not transmission. Other neglected zoonoses like Toxocara is very common in the southern part of the US, and the Centers for Disease Control have done incredible studies looking at the importance of toxocariasis, and they record ocular cases, and many of the ophthalmological centers are now collaborating to collect disease information on the importance of this important zoonosis. So yes, through education, CE was eliminated from the US. It was first eliminated from Iceland. In Iceland, they have long, dark winters, and a Danish writer saw that they had in the winter the Bible, and he wrote a book on control of Echinococcus. And then it combined this with legislation of slaughtering sheep under a year of age, and that's before the prediscolices developed, and Iceland became the first country to eliminate CE. New Zealand, many island nations have been quite successful. Cyprus, in Australia they have been fairly successful, but they have a macropod cycle between dingoes and wallabies and kangaroos and wild pigs, which keep the cycle going. So it's quite complex. Where you get wildlife cycles involved, you do have a reservoir that perpetuates the cycle when you control the domestic cycle. And in the US, there was no wildlife cycle in the contiguous US.
Okay, thank you. Our next question in the queue comes from Ariel, and let's give this a shot again. Ariel, I'm going to unmute you. And on your side, just hit the little microphone button, and we should have you on. Just chatted Ariel in the chat, so Ariel Archibald, you there? Maybe Ariel, you can type your question if the microphone's not working. And while Ariel does that, the queue is empty right now. So if someone wants to raise their hand or type something or unmute their microphone, now is a good time.
Maybe in the meantime, what we can do is maybe think about the need for this collaboration between the different disciplines that you mentioned. We had physicians, we had veterinarians, we had radiologists, we had nurses, anesthetists, obstetrics, gynecologists. How how can this experience in Africa be translated elsewhere? Because it is this type of partnership is what is needed for highlighted disease and many other of the neglected tropical diseases that Africa faces. But many of these diseases also reside elsewhere. Can you see this model of One Health partnership being replicated elsewhere?
I think in many parts of the world, the One Health partnership is flourishing and thriving very nicely. I think in countries where you have good wildlife populations, it's really good to get the wildlife folks involved and the climatologists, the people who do all the IT today, GPSing cases, and we still haven't really begun to have molecular markers for environmental contamination. Exactly how do people get infected? These are questions that we need good molecular scientists, environmental scientists and studies to really help with social scientists. So it's going to take a lot of people coming together to study the dynamics of many of these infections before we fully understand. With the Echinococcus cysts, they're very difficult to treat. And even though there are new puncture, aspiration, incubation, and reaspiration surgical techniques, advances, there are some stages. 3B on the ultrasound classification is incredibly resistant to chemotherapy, to all types of treatment. And we just don't know how these parasites can survive, no matter what we throw at them. So there are still many challenges. New therapies are required still.
Okay. We have the question coming in through the chat, so I'll ask it. And the question was, what is the prevalence of CHD in the Caribbean?
Well, the answer to that is no one has ever found a case of CHD in the Caribbean, which is quite interesting, and why not? We do have many sheep and goat farms, small holdings. The conditions are conducive to the parasite being here, but it's just never established, and to my knowledge there's never been a single case of CHD diagnosed in the Caribbean from somebody who hasn't had the infection possibility from elsewhere, which is really interesting. So, because it's very common in Latin America, and you know, Argentina, Chile, Uruguay, Brazil all have large numbers of cases of the Echinococcus. So I don't know.
How maybe have you been able to avoid it? Because you worked intimately with this disease for many decades.
That's an interesting question, and all my colleagues.
Yeah.
Actually, as the director of the program, that was my greatest fear, that one would wake up, you know, 2 years later, and either yourself or one of your colleagues would be diagnosed with a cyst in the brain, or a cyst in the heart, or a cyst in the pelvis, which, you know, is untreatable. It just disintegrates your pelvic bones. So it's a really nasty disease, and yes, we must have been exposed to an awful lot of challenges. So that's another, why do some people get infected? And I think this comes back to the whole seasonal droughts where people are immunocompromised just at a time when the dogs have the greatest numbers of infection. I think this must be a hard disease to catch. Certainly we would have been exposed. We're in contact with thousands of infected animals.
And you would have taken all of the infected contents from slaughtered animals, etc. And how would you have processed that content, disposed of it?
Well, that part we don't mind about, because you can eat all the cysts you want. As humans, we don't get anything
But
from
for the
that.
animals,
But it would have to be fed to the dogs, and then the dogs would produce eggs. So, but no, it was. I remember waking up more than once in a pool of sweat, thinking, "Oh my goodness, my colleagues would have this disease sooner or later." But thankfully, no one got infected, to my knowledge.
Thank you. Yeah, the next question comes from Brian. It's, are there species of dogs that have been found to be more susceptible to CHD than other species?
That's a good question, because there are the Basenji type of dog that is very common in the Turkana is very susceptible to infection with this parasite. Related parasite species like Echinococcus multilocularis can go into cats. We tried to infect hyenas, and hyenas are more closely related to cats than to dogs, and we couldn't infect hyenas. So there is presumably a perfect definitive host. Certainly most dogs, we don't know about different breeds, but all dogs are one species, Canis familiaris. Whether they're a Great Dane or a miniature Chihuahua, they can interbreed with difficulty, I guess. But so and dingoes, jackals, or wolves, all can get infected with this parasite. But cats are refractory to granulosis, but multilocularis, which if you look under the microscope, they're almost identical, doesn't infect dogs as well. It does go into dogs, but it's not as good as granulosis. So I presume there are species or breeds of dog, but remember that to be susceptible or to have the opportunity for infection, the dogs would have to be fed hydatid cysts with protoscolices. Most dogs won't get that opportunity. So for most dogs, pet dogs aren't fed uncooked offal, so they would never be exposed to infection. So we don't have that data, to be very honest.
Okay. The next question is coming from Richard in the chat box, and the question is, how does Richard get support to fight against cholera in my country and other African countries? So he hasn't said what country he's from, but presumably an African country. So his country and other African countries.
Okay, well, we know that sadly cholera rears its head
Ghana.
even more frequently today. Funding is a challenge. Though having said that, there is a lot more donors around today than historically. Very large philanthropic groups are challenging many of the diseases transmitted through poor sanitation, poor hygiene, such as cholera. Bill and Melinda Gates Foundation, and there are many other philanthropic foundations. The good news is that about 98% of all these charitable trusts that are looking for people to donate money to are on the web, on the internet. So there is money out there to be found. The question then becomes, how do you access it? How do you have the say, One Health team of experts that would help you access these funds that are there. You need a certain credibility. They're not going to give over dollars to anyone who asks. So there has to be some track record, some association with an established institution. But once you put the whole team together, in my own experience, the starting up a new foundation, it takes years to then start attracting donors who are going to be more and more comfortable giving you their funds once you have a team in place and a track record of utilizing funds in a good way. So the answer to the question of how would you get funding for cholera in Ghana is to have a team of people with good ideas who, and then identify the donors who are at institutions that are respected. And I know in Ghana there's a lot of institutions, and if you're part of those institutions, part of that group, and you get your team and you have novel ideas and hypotheses, novel ways of combating cholera. Cholera is a good example, actually, of what will become, or what continues to be, a serious problem. And to prevent cholera is quite easy. You need good water, you need good sanitation. And so you need good infrastructure. And if you don't have those things, then cholera can very quickly become very common. And it's not unlike many other bacterial, viral diseases that can spread very quickly in populations. So I think you just need to get together with good mentorship, good institutions, and apply for those funds which are out there.
And from the perspective of the one world approach, a disease that is emerging in one part of the world is not confined to that location only. In fact, from the African perspective, diseases that emerge in Africa is a global problem. It's not an African continent problem only anymore. So while we are focusing our conversation on the theme of the presentation on the African continent, recognizing from the one world perspective of the course that we're offering here right now, any disease in any part of the world does have global relevance because of the movements of people, the internationalism of travel, trade, industry, commerce as well.
Thank you. Yeah. So we have a series of questions now coming up. There are three in the queue, and the first one's coming from Matilda. And Matilda asks, is there any form of prophylaxis treatment for people who are at risk, i.e. immunocompromised? Could the albendazole be given as prophylaxis for people traveling to endemic areas?
Right. I think, Matilda, I think this is a very difficult disease to get infected with. Your chances of getting infected if you are a traveler to an endemic area, this is not one of those infections like malaria or yellow fever or dengue, Zika, chikungunya that you could be exposed to. And you need to take, in the case of malaria, prophylactic treatment. So there would be no need to guard against this disease. What I would say, if you're going to endemic areas, do not play with stray dogs. Do not pet dogs. These are, besides the threat of echinococcus, you know, you've got 64 other things that we share with dogs, like rabies. And probably the least parasite you should be worrying about is echinococcus. I think it's very hard to get it. You'd be very, very unlucky to be exposed with this parasite. It is a parasite that you have to live, I think, for years and have huge risk and transmission opportunities on an hourly basis to be infected. One of the interesting things is you probably ingest tens of thousands of eggs, and yet only one develops into a cyst. Most of the patients that we ever saw had a single cyst. So why does only one cyst become established? And that's one of the interesting things about the disease. But I would certainly not worry about this disease as a traveler. Stay away from stray dogs, and you know, plenty of bottled water. It'll prevent you from getting many other things that you might get in such endemic areas.
So basic sanitation hygiene applies across the board.
Across the board. Always washing your hands if you've been, if you do pet stray dogs, friends' dogs, you don't know, you know, wash your hands before you eat anything.
Yeah. Yep. Second question comes from Jan, and the question is, can they use anthelmintics in the dogs to prevent human contamination?
Absolutely. In fact, the control programs, what we were doing is you can use a drug called praziquantel, which is very effective at removing the adult tapeworm. And you would need to dose fairly regularly in areas where dogs might be used for herding, and they get hold of dead sheep that have died in the hills. In places like Wales and Scotland, where this parasite occurs, they have to keep treating dogs fairly regularly with anthelmintics. Praziquantel, which was developed by Bayer, comes in biscuit forms, all sorts of formulations, and so can be fed to dogs that are at risk. Now, a dog can only get infected by eating these protoscolices, so they can't get infected from normal canned food and drinking water from a tap.
Excellent. The next question, and the last one in the queue right now, comes from Yusuf. The question is, how feasible is One Health approach in combating echinocosis or any other neglected tropical diseases in terms of policy making and implementation? Knowing very well neglected tropical diseases cause high mortality in Africa, how can we make funders support a project that can tackle or reduce this mortality to the barest minimum levels?
Right, that's a good question, Yusuf, and I think that donors are much more aware, just as Dr. Badesi said, it is a global village today, and what might not be your problem might be a problem for your citizens. If you're from California and you happen to have a holiday in an endemic area, you can be at risk. I'm not saying you're going to get infected or take prophylactic drugs, but the world is ever more moving. People are wanting to explore the planet that we inhabit in ever greater numbers, and diseases can move. Dogs are taken all over the world as service dogs. After earthquakes, they might visit Turkey and might get infected whilst they're there, and they go back to their home countries. And so there are ways that the international community need to try and reduce the importance of these neglected diseases wherever they exist. And we have the tools today to do it. We have the prophylactic drugs, we have the treatments. We know that this disease is transmitted where you don't have careful slaughter facilities. So when people slaughter at home, and they might feed sister dogs, so you can get transmission. So by improving the totality of One Health, abattoir, slaughter facilities, good hygiene, good treatment of dogs, proper control of dogs, education, good hygiene, good you know, the key of it all is good education. I think you can travel, and I have the privilege of traveling to many parts of the world every year, and if you know how to avoid a lot of things, you can go anywhere in terms of disease problems. So I think education is, as Mandela said, the most powerful weapon with which you can fight these things.
And also from the perspective of policy, because how do we translate the One Health evidence, the One Health practices into policy? I believe that is where we think about health systems approach. We just spoke about education. We spoke about the slaughterhouses. We spoke about the animal production part of it. The point is that if we keep thinking in silos, as government systems are set up, different ministries of health, agriculture, education, environment, and if these ministries are not communicating with one another, if there is no cross collaboration across disciplines, that's a challenge. But the One Health mechanism allows for that opportunity to be able to share, to be able to explore, investigate, and then allow that evidence to inform policy decision making. So it is very critical to allow for the scientific efforts in One Health to be accepted and adopted by those in authority, the politicians, the decision makers, as well at the same time.
Well, Kanikokas exemplifies that.
Yeah.
If you try and get money to build slaughterhouses or to treat dogs, it is very difficult. Yeah. If you have a human problem and you can show that your citizens have this very nasty cystic disease, funding is a lot easier. So so that means that the veterinarians, the agricultural people need to work closely with the physicians, and ministries of agriculture, public health and health need to work very closely to obtain the funding, to leverage the problem of an economic problem in animals and a problem in people, and to maximize what a zoonotic infection is. Quite often, in fact, all too often, it is only the human importance of a disease is taken into account. And yet millions of dollars are lost by farmers with the condemnation of offal, which is an important part of an animal. And if they only have one or two animals and they lose the viscera, because they're condemned due to hydatid disease, that's an important loss to that family. So there needs to be cooperation across the sectors, and that's how, using One Health, you improve the funding opportunity.
And I think that was a good one to start wrapping up on. So we have about 10 minutes left. I'll give it over to Dr. Macpherson and Dr. Badesi for any kind of closing thoughts, and then Dr. Badesi, you can wrap up the session.
Well, it's been a pleasure to share my passion for 40 years of working with this incredibly complex disease. And if I was to say, looking back 40 years ago, the things that I didn't know I was getting myself in for, or for the journey I was going to take. So if I was back there then, what I would like to know, I'd like to know quite a number of things. One, I chose, quite by chance, a disease with global distribution. It really did facilitate my desire to travel internationally. I was sent, when we started the control program, to other control areas to learn how they had implemented their control programs. I would have liked to have known the holistic picture of this disease a little bit better than I did when I started. You learn incrementally throughout your career. At every turn, you pick up something new from another group. What I was particularly proud of and pleased about, and facilitated everything I did, was working very closely with surgeons and veterinarians and zoologists and biologists and mathematicians and biochemists, looking at the strain differences. And by working through the various collaborations, at one time in Nairobi, my lab was at the Wellcome Trust Labs at Kenyatta Hospital. My office was in the Flying Doctors. My experimental animals were out at Capetown at the vet school. Others were on farms in northern Kenya. And just the ability to work with so many different people has just been an absolute joy. So if I would say what my pleasure from One Health approach has been is the diversity of opinions and the expertise that if you work in isolation, and it's not good to work in isolation, partnerships is the way to go. You will have a fantastic opportunity of learning from different perspectives, and it was having the mentors that I had which enabled me to see that yes, I needed sociologists, I needed people who understood human behavior a lot better than I did to think about how to design questionnaires to look at the human behavioral aspects of the disease. We still have many questions to answer. It's still a great disease. It's still, unfortunately, out there. And it will still take a lot more people studying it to control it. The other thing that I have enjoyed very much is the society of echinococonuts, as we're called, is quite small. And so you quickly get to know people in 30, 40, 50 different countries, all sharing the same passion of trying to work out how to get rid of this disease. And that has been a great pleasure and a privilege of working in this field.
Well, thank you. Thank you for affording us the opportunity to learn and glean from your experiences as well, not only in terms of echinococcus, but also in terms of your own international health work and contributions you would have made over time as well. I know that as a student of parasitology in my time, I remember learning a lot of parasitology indirectly through you, where a lot of our specimens were labeled CNL Macpherson. I was wondering who that person was, but I have the absolute privilege now of working with CNL Macpherson as well. So here we are in March of 2017, and we will be continuing to share the year that is 2017 as we continue our course in the month of April and each month throughout the year. Coming up in April, we have one of our brilliant dual degree students who is studying veterinary medicine and public health, also going to share global perspective as we continue to engage health, not only in terms of the human aspect, the animal aspect, and the environmental aspect, but throughout all regions of this planet that we reside. So thank you for joining us. Look forward to having you with us again for our next installment of One World, One Health, One Medicine. Please ensure to enroll in the course, enroll at online.stu.edu/anniversary. I'd like to thank John Soap, who supported us in developing this course, but also who is always at hand as well in coordinating these live sessions as well. Please note that apart from the live session, this session is recorded, and we also have some discussion questions and a short, simple, and easy quiz as well to ensure that we engage the topic and the content from an academic perspective as well. So it is goodbye for now. Continue to enjoy the month of March, and we will see you guys back in April for another One World, One Health, One Medicine series. Goodbye for now.
Thank you.
Thanks. Pleasure. Thank you. Yeah. Okay. Yep. That's good.
Thank you, John.